Hello, my name is Sarah Brookman, and today we are going to be talking about scopolamine, also known as the devil's breath. Scopolamine can be either naturally or synthetically produced, and it's made from the combination of tropine alkaloid and anticholinergic drugs. Originally, scopolamine was used to treat motion sickness, nausea, and vomiting. It has also been used to reduce the production of saliva during surgery requiring anesthesia. The source of scopolamine is called burundanga, and it has similar effects to a roofie, except this drug makes individuals highly suggestible while being conscious and able to communicate. This drug is extremely deadly and is often used to make people suggestible by people intending to commit crimes. It has even been called the perfect substance for criminal acts. Following taking the drug, people have total memory loss for the time that they were on the drug. Scopolamine is a fine white powder and is often mistaken for cocaine by appearance. Scopolamine can be obtained from plants belonging to the solanase family and is a secondary metabolite of these plants. Overdoses of scopolamine can lead to delirium, delusions, increased body temperature, and even death. Scopolamine can be administered transdermally, through a patch, orally, subcutaneously, intravenously, or optimically, meaning through the eyes. Though uncommon, scopolamine can be used recreationally in conjunction with opioids to increase euphoria, or even less commonly, used on its own to produce hallucinogenic effects. Most commonly, though, it is used for interrogation, being tagged the truth drug, and for criminal use, as previously mentioned. Scopolamine can be used as a drug of abuse, most commonly after the use of transdermal patches, and withdrawal symptoms include headache, vomiting, and nausea. Hospitalization is sometimes required in severe cases of withdrawal. Now, let's talk about the neuroanatomy of scopolamine. Through the use of cytochrome C oxidase, scopolamine was traced as it relates to memory impairment in rats. This study, conducted by Hersham et al. In 2014, found a principal concept that the effects of scopolamine are not limited just to cognitive parameters, and that when rats were given scopolamine, the levels of choline acetyltransferase was reduced within both the amygdala and the CA1, which is the first region within the hippocampal circuit. Other implicated brain regions include the nucleus basalis, primary motor cortex, and globus pallidus. Additionally, the injection of scopolamine reduced the number of C-FOS cells within the amygdala, and cytochrome C oxidase expression was reduced in regions responsible for movement, memory, and anxiety. Overall, this study suggests both locomotor and anxiety-related behavioral effects of scopolamine. Scopolamine affects the neurotransmitter acetylcholine, which is responsible for regulation of attention, and is a muscarinic antagonist. In a study conducted by Fneal et al. In 2009, 12 healthy volunteers were recruited to perform the attention network task after given zero point 0.4 milligrams of scopolamine. It was concluded that compared to the placebo group, those given scopolamine increased in reaction times for conflicting stimulus processing, as well as had a decreased activation in the anterior cingulate cortex, which plays a role in conflict processing. Scopolamine is a tertiary amine that acts as a muscarinic antagonist that affects the executive control of attention, as well as reduces brain activity within the left superior and left middle frontal brain areas. On a neuropsychological front, scopolamine selectively affects verbal learning and memory, but does not seem to affect aspects such as planning and working memory. Overall, this study found muscarinic modulation of dopaminergic neurotransmission in frontal attention networks while processing conflicting information. In a study conducted by Green et al. In 2012, it was found through a multimodal approach that scopolamine antagonizes muscarinic type 1 and histamine type 1 receptors within the central nervous system, hypothalamus, and vomiting center of the brain. Here, it is visualized what direct antagonism looks like. Now, how does scopolamine relate to neurocircuitry? Well, as mentioned previously, scopolamine is a muscarinic antagonist, which means it is blocking the binding of the neurotransmitter acetylcholine. We also mentioned that the role of acetylcholine is related to cognition and memory, and if acetylcholine neurons are not functioning properly, then learning and memory dysfunctions can occur. Acetylcholine is also important for pain, attention, mood, and sleep. Scopolamine antagonizes acetylcholine receptors within the central nervous system, and as such is an effective amnesiac agent. Now, it is important to note that acetylcholine has two types of receptors, nicotinic, which are ionotropic receptors, and muscarinic receptors, named M1 through M5, which are metabotropic receptors. Unlike many other neurotransmitters, acetylcholine does not have a transporter, but instead has its action terminated by the enzyme acetylcholinesterase. Acetylcholine pathways begin in the basal forebrain, specifically the nucleus bacillus and the medial septal nucleus, a nucleus of the diagonal band. These nuclei project to the fornix, cerebrum, and hippocampus. The pedunculopontine nucleus and lateral dorsal tegmental nucleus send acetylcholine projections down the spinal cord and to the cerebellum. So how does scopolamine fit into all of this? Scopolamine is involved in both acetylcholine and dopamine transmission. As mentioned previously, scopolamine binds nonspecifically to the M5 muscarinic acetylcholine receptor throughout all regions of the brain. In a homeostatic state, the M2 and M4 receptor subtypes of the acetylcholine receptor are linked to a GI protein found in the neuronal terminal of the mesopontine cholinergic neurons and inhibit the formation of cyclic AMP. These are mostly presynaptic autoreceptors, and antagonizing M2 or M4 receptors increase the release of acetylcholine and activates both muscarinic and nicotinic acetylcholine receptor signal transduction cascades. M1, M3, and M5 acetylcholine receptors are mostly GQ coupled. These are autoreceptors that generate a negative feedback in order to decrease muscarinic acetylcholine signaling. However, when scopolamine is in the body, this negative feedback is blocked, which disinhibits cholinergic transmission. Elevated dopamine release can result from increased acetylcholine release into the postsynaptic neurons of mesopontine. It should be noted that these neurons are primarily dopaminergic neurons within the ventral tegmental area or substantia nigra. Additionally, scopolamine influences theta, delta, alpha, and beta activity in electroencephalograms. This leads to memory deficits that are similarly expressed to those present in elderly populations. As mentioned previously, scopolamine is a non-selective competitive inhibitor of M1 through M5 receptors in the central nervous system. The exact mechanisms of scopolamine are not yet entirely understood. However, emerging research has pointed to the role of the M1 and maybe also the M2 acetylcholine receptor antagonism at inner neurons, which inhibits the release of downstream neurotransmitters. This additionally causes the release of pyramidal neuron activation in order to regulate neurological responses related to depression and stress. Scopolamine also antagonizes M4 and M5 receptors, which can benefit disorders such as schizophrenia and substance use. The antagonism of the M1 through M5 receptors does not account for the ability of scopolamine to prevent nausea and vomiting, but does account for its anticholinergic effects and the ability to alter signaling in the central nervous system. Now, let's talk about the implicated brain regions related to scopolamine. In a study conducted by Graysby et al. In 1995, upon the administration of scopolamine, there was decreased regional cerebral blood flow in the right thalamus and right and left lateral premotor areas. Moreover, the right and left prefrontal cortex and the right anterior cingulate were implicated in scopolamine's ability to attenuate memory task-induced increases of regional cerebral blood flow. Additionally, scopolamine reduced brain activity in the left superior and left middle frontal brain areas. So, what does all this mean, and why is it important to understanding the circuitry of scopolamine? Scopolamine decreases blood flow to the left and right thalamus. The thalamus plays a role in sensory motor signals as well as regulating functions associated with consciousness and alertness. If this region of the brain receives less regional cerebral blood flow, then sensory motor as well as consciousness will be reduced behaviorally. The left superior and left middle frontal brain areas are implicated in higher cognitive functions, and in particular, working memory. This can account for why people under the influence of scopolamine may experience a sort of hypnosis or extreme suggestibility. How does scopolamine relate to neuroplasticity? Scopolamine is a non-selective muscarinic antagonist that has downstream effects linked to neuroplasticity within glutamatergic synapses and the resulting antidepressant effects. These antidepressant effects can be attributed to the antagonism of the M1 receptor. When the M1 receptors are blocked in the brain as a result of scopolamine, there is a transient disinhibition of glutamate release, and this creates neuroplasticity at the glutamatergic synapse. This means that there is a short-term selective and transient reduction of synaptic inhibition of glutamatergic projection neurons due to GABA interneurons firing. The firing of cortical GABA interneurons leads to the tonic inhibition of glutamate release. These cellular effects converge on the AMPA activation pathway. This is mediated by the transient increases in tonic glutamate release. So, what does this mean long term, and why is it significant? We know that scopolamine is an antagonist for the M1 muscarinic acetylcholine receptor, and this leads to tonic glutamate inhibition. It is important to additionally note that the M1 agonist can enhance cognition. So, if the inverse occurs and the M1 receptor is disallowed to open, then, behaviorally, this can lead to cognitive impairments. This is backed up by research showing that scopolamine administration results in cognitive deficits. Additionally, increases in tonic ambient glutamate can saturate NMDA receptor activation and desensitize NMDA, AMPA, and kainate glutamatergic receptors. This can lead to challenges with information processing at the neuronal level, as these receptors are important for memory formation. NMDA is important for plasticity, specifically long-term potentiation. If scopolamine is desensitizing NMDA, memory formation can be prevented. The long-lasting effects of scopolamine are thought to increase synaptic plasticity. Specifically, synaptic proteins reorganize synaptic connectivity, and these changes remain for a significant time after the drug has left the body. So, how does scopolamine relate to pharmacology? Scopolamine has an average half-life of 9.5 hours, though this can vary depending on the route of administration, and scopolamine can readily pass through the skin. If transdermal patches are used, the effects of scopolamine can last for up to 3 days. Scopolamine can cross through the placenta and the blood-brain barrier. It is also hypothesized that it reversibly binds to plasma proteins. Scopolamine is a competitive inhibitor for muscarinic receptors for acetylcholine and is a non-selective muscarinic antagonist for M1 through M5 receptors. Scopolamine has dose-dependent effects. The transdermal patch is used to measure plasma concentrations over longer periods of time in order to negate this. Upon the application of the patch, scopolamine can be detected after 4 hours and reaches peak concentrations at 24 hours. Scopolamine is primarily metabolized in the liver, and the primary metabolites are sulfide conjugates and different glucuronides. The half-life of scopolamine differs predicated on the route of administration. Intravenous administration is roughly 68.7 minutes, oral is roughly 63.7 minutes, intramuscular is roughly 69.1 minutes, and a transdermal patch is roughly 9.5 hours. Moreover, scopolamine is somewhat soluble in water and soluble in fat. This allows it to pass through biological membranes. So, how does scopolamine act on receptors? Scopolamine is thought to decrease NMDAR gene expression, and this affects NMDA modulators, including ketamine. This can prevent glutamate neurotoxicity. Moreover, scopolamine induces the mTOR pathway, the central regulator of metabolism in mammals. Scopolamine also increases levels of BDNF, an immediate early gene that promotes synaptic plasticity, specifically long-term potentiation, and the formation of dendritic spines. This can tie back to our previous discussion on neuroplasticity. Additionally, scopolamine has been used to treat major depressive disorder as it is an antagonist for the M1 muscarinic receptor. On an entirely different note, scopolamine also has non-muscarinic effects. Small concentrations of scopolamine induce depolarization alongside decreased cell input resistance. It is shown to increase membrane permeability for both sodium and potassium. This process is thought to be entirely independent of muscarinic cholinergic receptors. Now, let's talk about the research methods that yielded this data. A primary research method used to pinpoint the functions of scopolamine in the brain is using knockout mice. For instance, the antidepressant effects of scopolamine were explored in 2 different studies using M1 knockout mice. By knocking out the M1 muscarinic acetylcholine receptor, researchers observed that the antidepressant effects of scopolamine were no longer present. It could be deduced from this that the M1 receptors are responsible for scopolamine's antidepressant effects. Transdermal patches are also heavily used in scopolamine research, as it allows the drug to be absorbed at a constant rate and to measure plasma concentrations over time. A common research method used is microdialysis. Specifically, a study by Wei et al. In 2014 used differing routes of administration, including subcutaneous, oral, and intranasal, to assess the concentration of scopolamine in plasma, specifically within the olfactory bulb and vestibule. The result of the study was that scopolamine can be directly absorbed intranasally into the olfactory bulb. Microdialysis involves the implantation of a cannula directly above the brain region of interest and inserting artificial cerebrospinal fluid in order to have the neurotransmitter diffuse into the efflux. This is the most often used in the study of scopolamine to assess routes of administration as well as neuroanatomy. Another major research method that I encountered during this project was the use of adeno-associated viruses, or AAVs, to over or underexpress genes in order to change behavior. In a study conducted by Liu et al. In 2021, AAVs were used to knock down the M2 muscarinic acetylcholine receptor in the medial prefrontal cortex. The researchers found that this M2 receptor plays a role in the antidepressant effects of scopolamine. This study, as did others I encountered, employed Western blotting to assess protein detection and concentration. The final major research method that I encountered was immunohistochemistry. Immunohistochemistry is used to assess protein expression. This method was often employed in studies connecting scopolamine to impairments in cognitive functioning, including Alzheimer's. Genes such as CREB1, BDNF, and TRKB are associated with cognitive decline as it relates to scopolamine, and as such were assessed during immunohistochemistry studies of scopolamine. Here are all the references that I used for this project. Thank you so much for watching.